Hello everyone, welcome to ENIO's webinar. First, let me briefly introduce myself. My name is Konstantinos Boutetakis and I'm a biologist, bioinformatician at the National Hellenic Research Foundation at the Institute of Chemical Biology, and member of ENIO's Bioinformatics spin-off company. On March 2013, Elizabeth Payne in Science magazine posted that someday it should be possible for doctors to send individual cancer patients in for a genomic analysis and based on their results, prescribe the drug they know will be the most effective. Now, the time has come, and bioinformatics has played an increasingly important role in this evolution. In the first part of the webinar, I'm going to talk to you about how we can retrieve genomic, specifically RNA and clinical data from the Cancer Genome Atlas program, well known as TCGA, using programming language in our studio environment, and how we can derive a gene signature via Bioinformatics platform developed by ENIO's company, with genes that are potential biomarkers for diagnostics, drug targeting, and other applications that rely on the discovery of strong molecular phenotype associations. The transcriptional profile of this gene signature in cancer patients will help us stratify the tumors into different subtypes, evaluating its discriminating and prognostic power with respect to the overall survival rate. In order to execute the appropriate commands in our studio, please install and load the following libraries into your environment. Of course, before that, please be sure that you have already installed app of version 4.0 or greater and Bioconductor of version 3.11. The first packages summarized experiment, TCGA BioS, and TESA are bioconductor packages, while the last two, DeepLear and DT, are included in a comprehensive archive network known as CRAN. Of course, you can check their documentation for more information, a video of our studio, or on the web. Bioconductor is an open source of tools for the analysis and comprehension of high-throughput genomic data. It uses for statistical language, and it has two releases each year and an active user community. You can install the stable version of Bioconductor packages, typing this code into your studio console, as you can see here. At this point, let me give you in brief some general information about the TCGA consortium. It's an NIH initiative that makes publicly available molecular and clinical information for more than 33 types of human cancers, including exome data, SNPs, DNA methylation data, transcriptome, microRNA, and data. The subtypes available at TCGA are the primary tumors, the recurrent solid tumors, the blood normal and tumor cells, the metastatic, and the solid tissue normal cells. All this data is accessed via three main sources: the CDC data portal, the CDC archive, and the Broad Institute CDAC Firehouse. The first one, the CDC data portal, provides access to the subset of this data that has been analyzed against current human genome assembly. And we will gain access to it via RNA packets, which is called TCGA. So as to query, we will focus on the head and neck squamous carcinoma study defined as TCGA HNST project in this project. You can have a look at the available cancer projects in CDC with the execution of this command here with the get GDC project function, assigning the result to a data frame with the name, for example, GDC projects, while the base function head will print the top 6 project IDs with their names in console. Another useful function of TCGA BioLinks is the get project summary, which returns a summary of the data available in GDC portal for every project. You can give it a try with our project, TCGA agency, and yeah. Let's begin our analysis by querying the GDC portal for RNA-seq transcriptome data regarding our project. The first action of TCGA BioLinks we need to make our query is the GDC query, and we request GDC portal to search for deposited RNA-seq transcriptome data in 8-sec count summarized at gene level, assigning accordingly the arguments as you can see here. In case our study is valid and are available data for downloading, we can list the candidate tables with the use of getResults function, giving as query argument our query data frame, and as codes argument or column argument the cases. The tables that is, the tissue type, such as solid tissue, normal, primary, soft tissue, cell line, etc., regarding our query can also be returned by key results, giving us cause argument the subtype value, as you can see here at the bottom of the slide. You can take a look at the columns of data returned by get results. If you as input the get results output in the call names function. You can try it. In our case, we are not going to use all tumor samples of this cohort, but we focus only on the comparison of tumor samples with their paired tissue specimens. This allows us to not only reduce the bias coming from different sample sources, taking the extremely small number of normal samples compared to primary tumors, but also to do our results more comparable. For this reason, we have to filter the subs data list for matched primary solid tumors and solid tissue normal samples, defined by the TP and N steps respectively. This can be done by the TGA query math couple sample types function, giving the subs argument as depicted here. Now we can refine our query, searching only for data regarding the cases which include in a paired SM list. These samples are assigned to barcode argument in GDC function. Please be patient because this task might take a while. It depends on the size and number of files to download and your real connection. If downloading failed for one or more files, you can rerun the previous command until no error message is generated. The files that have been fully downloaded will not be downloaded again. All the requested data, as they are described in the query down data frame, can then be downloaded and saved in the folder with the GDC download function. The name of folder, GDC data, is system defined and it's created under your working directory following hierarchical file structure. It's very important to note that your working directory must include the GDC data folder. You can check this at the lower right-hand window of the RStudio interface, where your working directory is displayed. Now it's time to create an R object called summarize experiment that contains all the information we have retrieved by GDC portal. This can be done with the third main function of TCGA Bioinks, which is the GDC prepare function that reads the downloaded data and prepare it into a summarized experiment object. It's recommended to save this object locally to your working directory to an RDA file so as to load whenever you want with the working RDA. As you can see here, I have already saved this object in my working environment with this name. Regarding summarized experiment, I would like just to mention that it's a matrix-like container which contains one or more assays, each represented by a matrix-like object of numerical mode. The rows represent different ranges or features of interest, like genes, transcript sections, and represent tables. Using the command summarized experiment in your console, you will find information about this topic. And a very useful serial library for testing is the TD, because you can fetch the clinical and expression data of your summarized experiment object in a pop-up window with scroll bars displayed states on HTML pages. Its data types function, as you can see here, provides filtering, pagination, sorting, and many other features in these two different tables. However, except this useful function of the library that permits better visualization of data, there are also many functions that give us access to the different parts of a summarized experiment object: core data, raw data, and assays. The first one, the core data function, the clinical index data associated with our cases. For more investigation of clinical data, you can also create a frame with the clinical information in your RStudio environment with execution of this command. You will return the output of core data function to the basic function called names. We get important information regarding the subclinical attributes. Any attribute of this can be used as a filter to clean our data from undesirable cases. For our study, we will use the is FFPE to remove formally fixed and paraffin samples known as FFPE samples because of the high degree of nucleic acid degradation in FFPE tissue. While nucleic acids can be isolated from the type of preserved tissue and can be of limited use. Therefore, FFPE tissue may not always be suitable for molecular genetic analysis. Moreover, most of the FFPE protocols are not standardized. There is no gold standard. All of these can disproportionately affect the results gained from them. especially in the case of molecular genetic analysis. Let's check the number of FFP samples with the table function, as you can see here. Provide a small summary with the total number of different states of each. In our case, no samples FFP, so our data does not need filtering. Otherwise, you should exit to clear off. But what about gene expression data access? How can we access the read counts in HDC format for our cases? We can use the assay function to access the HDC count matrices and draw data function to see the gene mapping information. You can take a look at the first 6 records of these tables with the base function head, as you can see here. Well, that's about it for this part. We have covered the section, how can we access genomic and clinical TCGA data using the R programming language and the powerful library TCGA BioS. I'd now like to move on to the next section regarding the correct identification of differentially expressed genes, division, between cancer and normal cells, which is a key in the understanding of phenotypic variation. This procedure is well known as differential expression analysis. The number of methods and software for differential expression analysis for RNA data has been increased rapidly over the past few years. However, there is no consensus about the most appropriate pipeline. The first crucial step of this procedure is pre-processing. We identify samples with a low correlation gene expression distribution and identify the possible outliers and should be filtered out from further analysis. A useful way to do that is the TCGA analyze pre-processing function of TCGA BioS package. You can define a square symmetric matrix of Spearman correlation based on the RNA count distribution, producing also a box plot of subcorrelation. You can use Spearman correlation defined by the co.cat argument to filter the samples. The default value is 0 when there is no filtering, but the value of 0.6 is recommended for reliable results. You can take the output into your working directory, of course, as a PNG image file with a name in the file name argument. Our sample list does not include outliers, so we can proceed to the next step, which is the normalization step. Take into your consideration that the read counts in this raw format may have biases because of intrinsic artifacts such effects. Variability measurement can be attributed to biological and technical factors, and we care only for biological variation. Technical variability must be evaluated and be parameterized in our analysis. When we are talking about technical variation, we are referring to differences in library preparation of consoles, sequencing errors, mapping and fermentation bias, sequencing composition and similarity, the difference in length and sequencing depth. That is, how many read quantified sequences and many others. Such unwanted noises can significantly reduce the accuracy of statistical inferences from RNA-seq data, and also prevent research from properly modeling biological variation and group-specific changes in gene expression. Hence, thoughtful consideration is needed in determining how to normalize RNA-seq data. In our analysis, we will use the HR algorithm for identifying differentially expressions between normal and tumor cells. HR is one of the most widely used tools for differential expression analysis with linear zoom and dissect-dual algorithms as well. It has been shown to perform well in terms of precision, accuracy, and sensitivity, and you can find more information about this algorithm in the paper of Robinson, McCarthy, and Smith in 2010 in Bioinformatics journal. As a first step for adapting our data to HR is the variable setting, which means that we have to specify the clinical feature, summarize the experiment object that we will use to group our samples and divide them into 2 discrete classes. This clinical feature will be the type of tissue, defined as primary solid tumor and solid tissue normal respectively, which is available in the summarized experiment object with the use of call data function, using the columns definition, subtype, and soft letter code. However, HR requires group variable, so we have to convert it with the factor function, because we have to convert our group variable to a factor with defined levels. Okay, This can be done with a factor function. But this function sets the levels of a factor in lexicographic order by 49. So we can use also another function, the level function, as you can see at the bottom of this slide, to define the reference base level. In our case, we set it to normal samples that are symbolized by NG term.